Located in the nucleus of the cell is the genetic material of an organism, defined by the biological molecule deoxyribose nucleic acid, or DNA for short. The DNA molecule is often described as a double helix, or twisted ladder, and consists of three major components: deoxyribose, phosphate, and bases. There are four distinct bases in the DNA molecule: adenine, thymine, guanine, and cytosine. Adenine and guanine consist of two rings, a 6- and 5-membered rings, and are known as purines, while thymine and cytosine consist of 6-membered rings and are known as pyrimidines. Due to the polarity of the molecules, adenine and thymine pair with 2 hydrogen bonds, while guanine and cytosine pair with 3. These base pairs form the rungs of the ladder. The bases are covalently bonded to carbon atom 1 of a deoxyribose sugar. Deoxyribose is a pentose furanose. Pentose because it is a 5-carbon sugar, and furanose because it forms a 5-membered ring structure. The deoxyribose sugar is not exactly planar. One of the carbon atoms is aligned out of plane to reduce tension between the covalent bonds. This is known as a sugar pucker, and a pucker can either be endo when it is above the plane or exo when it is below the plane. A base and a deoxyribose sugar forms a nucleoside, and it becomes a nucleotide when the deoxyribose sugar forms a covalent bond with phosphate at carbon atom 5. The hydroxyl group at carbon 3 of deoxyribose covalently bonds with the phosphate group of another nucleotide. A chain of nucleotides forms a DNA strand, and the ends are denoted as 5' and 3', referring to the carbon atoms of deoxyribose with the phosphate and hydroxyl groups respectively. The bases of a DNA strand are read in the 5' to 3' direction. This directionality is due to the direction of DNA replication, which will be explained in another video. Additionally, the DNA strand pairs with a complementary DNA strand. The two DNA strands are anti-parallel to each other. Here's a DNA model that I have made for demonstration purposes. I would like to point out that the bases are facing you for easier presentation. They are actually flat and stacked on top of each other. While the bases do pair with each other via hydrogen bonds, the remaining regions of the bases are actually hydrophobic. As such, the DNA ladder would twist to minimize contact with the aqueous cellular environment. This gives the characteristic double helix structure of DNA. Helix because it spirals around itself, and double because of the two DNA strands. In a double helix, there are numerous physical parameters in a DNA molecule. There is a rise, which is the vertical gain between two adjacent bases. There is also the pitch, which is the vertical gain in a full helical turn. By dividing the pitch by the rise, you get the repeat number, which is the number of bases in a full helical turn. Additionally, the direction of the helical turn determines the handedness of a DNA double helix. A DNA is left-handed when it rises towards your left, and it is right-handed when it rises towards your right. With all the physical properties of the DNA that I have mentioned, there are 3 different forms of DNA: A, B, and Z.